thank you, Celeste, and for inviting me to give this talk. I must say that I first encountered Larry when I started as a postdoc with Charles Laird, who's over here, and I'd come from Matt Messelson's lab, and I worked on Drosophila heat shock. But actually, I was most interested in satellite DNA. Now, satellite DNA gets its name from the fact that in cesium chloride Messelson stall gradients, there's a main band, but there's also satellite bands that you see occasionally. And that's because they have a different buoyant density because they're homogeneous and have somewhat different buoyant density, different base composition from most DNA that's in the main band. And anyway, Matt was aware of the fact that in closely related species, you can have different satellite DNAs, and he thought that maybe this is responsible for speciation because you see it between closely related species. And he went so far as to talk Dick Lewontin into believing this, and he went ahead and decided that what he would do is go to fertility clinics, look for couples who were infertile and look at their satellite DNA and maybe find incipient species, and that was his idea. And but I thought it sounds crazy, but I thought it was a pretty good idea, and so I was interested in somehow getting into satellite DNA. So when I came to Charles's lab, he pointed me to the fact that there is this phenomenon called position effect variegation. I'd never heard of it before, but it's where genes get juxtaposed to heterochromatin, and then they show silencing, and I realized, I can combine my two interests in heat shock response and also heterochromatin, which is where the satellites lie. And of course, the guru of satellites at that time was Larry Sandler, and so I would go across the street. I was in the zoology department in Charles's lab, go across the street and interact with Scott and the other fly pushers there, but in particular Larry, who would love to talk about this stuff. And I sat in on his course, and I drank the Kool-Aid, and and this is the way I remember Larry. I don't know if he's making a point or actually there's a punchline of a joke or both probably. But anyway, I really got into it. And at one point, my position effect variegation on a heat shock puff project was going really well. And I told Larry, and he invited me to give a departmental seminar on it. And when he introduced me, he went through the whole history of position effect variegation, beginning with Muller in 1932. And everybody who started working in Drosophila genetics wanted to work on position effect variegation because they thought they could solve it, but never could. And what he then said was, so this is like sowing one's wild oats. It's something you got to get out of your system. You do it when you're young, and then you move on and do something useful. And I think Larry had it right there, because try as I could, that position effect project, I don't think it really told me much about position effect variegation. And I ended up only getting a job because I cloned fly genes in yeast with Ben Hall. But so actually, he was quite right about that. I didn't really solve position effect variegation at the time, but it did whet my interest in the idea that if you study heterochromatin and satellites and centromeres, you might be able to find a connection between them, and that leads to the question of what specifies a centromere. Now, this question is even older than position effect variegation, because you could have asked this question back just looking at these drawings from Walter Fleming, where you see from the inverted V shape of these chromosomes, their evident attachment to the mitotic spindle, and especially these heterochromatic knobs that Fleming drew on here. It was clear that there's one position present on every chromosome that's responsible for pulling the chromosomes to the pole. But what is it? So fast forward a century, and Louise Clark and John Carbon answered the question quite clearly. They took a 1.6 kilobase fragment of DNA, they cloned it in a replicating plasmid, and they showed that it could maintain segregation faithfully through meiosis and mitosis. Then they went on to show that if they look at all 16E centromeres, they could identify a consensus sequence, actually 3, that they called CD1, 2, and 3, and later work showed that CD1 is the binding site for the CBF1 general regulatory factor, and I'll say more about that much later. CD3 is the binding site for the kinetochore specific complex, CBF3 complex, that recruits a centromeric nucleosome over this 80 base pair region that's extraordinarily AT-rich. And as Sue Biggins has shown, that you have a centromere here with the centromeric histone, CSC4. We prefer to call them collectively centromere H3 to point out the fact that they're histone H3 variants. For purposes of this talk, I will call them centromer protein A or CEMP-A. Now, this gives me the opportunity to talk about, so what we were interested in doing was following up on Sue's work and actually determining just what is in this 80 base pairs. What is that particle? Nucleosome should be 150 base pairs, but this is much smaller. Now, so over the years, we've been focused on this question, and I'm just going to give you one bit of evidence that we think explains what's actually in here, and it gives me the opportunity to tell you about a method that I'll be referring to later, which we call cut and run. It goes like this. We mix live cells with lectin-coated magnetic beads. They stick. We permeabilize, add an antibody. The antibody diffuses in to find its target, say a transcription factor. We then add a fusion between protein A and micrococcal nuclease, MNase, which will bind, the protein A will bind to the IgG of the antibody, anchor the MNase. We add calcium to activate the enzyme. And when it cleaves on both sides, the factor can come out. We extract the DNA, do paired-end sequencing. It's one day from live cells to purified DNA, and the backgrounds are extraordinarily low, such that we and a lot of other people just use this instead of chromatin immunoprecipitation. Now, we're using the supernatant here. However, the yeast centromere is extraordinarily unstable, and that's because it's like a 10 megadalton complex that's bound to one site in the genome and renders it completely insoluble. But it turns out that we can use cut and run in order to look at this. So we split the sample, we extract the total DNA and the soluble DNA, and the difference is the insoluble DNA. So now when we apply this to the yeast centromere, CSC4, what we see is over 2 orders of magnitude range of digestion, the insoluble fraction is loaded with CSC4 and is directly 80 base pairs right over CD2 where it should be. We also did this with histone H2A, and we see the same thing despite the fact that there's H2A in every nucleosome on the entire chromosome, almost. The centromere is the centromeric histone, the centromeric nucleosome, is the most insoluble site on the chromosome. So it's been controversial as to whether the centromeric nucleosome in budding yeast has H2A, H2B dimers, but you can judge for yourself. Now, as Ginger pointed out, this is a point centromere, but there are many other types of centromeres. So, for example, fission yeast has a regional centromere that at first we thought has no distinguishing features and has CEMP-A, and then in the most homogeneous region for most plants and animals, including humans, you basically have the satellite that I referred to earlier that we were most intrigued by. However, it's not the only type. When Florian Steiner was a postdoc in the lab, he showed that actually C. elegans, which is holocentric, has these points, looks like point centromeres throughout, so they're actually polycentric. And most strikingly, when Anna Drindenberg was a postdoc with Harmeet Malik and myself, she showed that actually holocentric insects, including butterflies, for example, do not have the same machinery. They don't have centromere protein A or anything that we're accustomed to finding at centromeres. Then there are these curious neocentromeres, these human neocentromeres. So what you're looking at are chromosomes that, from an individual, in which we're marking the centromere and the satellite, alpha satellite here, with centromere protein B that I have a lot more to say about later. And what you can see is that these three chromosomes at the primary constriction, you have CEMP-A. However, in this chromosome, chromosome 4, there's been a shift from its normal position in alpha satellite to a new position more medially because, with CEMP-A there, because CEMP-A always goes with the active centromere. Now, the individual from whom these chromosomes came was actually phenotypically normal. In fact, this chromosome has undergone at least 3 human meiotic generations, so it's a normal centromere. And these kinds of observations have led to the idea that centromeres are epigenetic because there's no alpha satellite there, so what could sequence shouldn't matter. But there's a real problem when you think about it with centromeres being just epigenetic and sequence doesn't matter. And this gives me the opportunity to get to some background to what I'm going to tell you next. And that is this term meiotic drive that was introduced by Larry in 1957. And in this paper, they talk about segregation distortion as an example of meiotic drive. They didn't know at the time that actually meiosis is normal in this case. However, there's one example that they have here in which is really meiotic drive, and this comes from Marcus Rhodes, what he called preferential segregation. Now, Rhodes was studying knobs in corn, and these knobs in corn are distal heterochromatic elements that have unusual behavior in the presence of a particular knob called abnormal knob 10. It goes like this. If there's an odd number of recombinations between the centromere and the knob, what happens is that the knob will actually lead at anaphase 1 and will end up in a gamete, and the gamete's in a preferred position to make it to the next generation. Okay, now, when Harmeet was a postdoc in the lab, he discovered that CEMP-A in Drosophila was undergoing adaptive evolution, and we realized that this kind of implied that there's an arms race going on. But how can you have an arms race for something as essential as the basic centromere protein, which is essential for every cell division? Because in an arms race, there are going to be winners and losers, and the losers are just going to, will basically be unfit, and the same goes for male meiosis. However, what Rhodes was describing, this kind of process actually would work in the sense that if centromeres can play the same game, then what will happen is that it will end up in the preferred position, and this is true for both plants and animals. And so this led to the idea that what's going on is that the satellites are actually expanding in female meiosis. They're basically expanding, and in female meiosis, they will get more CEMP-A, so they will have more microtubule contacts, and therefore they might be able to reorient themselves so they would go to the favored pole. It's a completely selfish process. However, we also knew that from work showing that if you have an unbalanced segregation from Bruce McKee, if there's an unbalanced segregation, it could abort male meiosis, so it aborts spermatogenesis. So if this same thing happened in the male, it would be deleterious. But that allowed us to explain what could happen. So it's driving in female meiosis, but it's sterilizing males would be deleterious. However, if it goes far enough that it can encounter a mutation in centromere proteins such as CEMP-A, if it mutates such that it restores the balance parity between the two, then both the centromere and the mutant will drive the fixation together. And so this basically can explain what we refer to as the centromere paradox. That is, you have this rapidly evolving DNA, the thing that Matt Messelson first got excited about. In fact, we had actually pointed out that centromere drive is occurring in Robertsonian translocations, which are fusions between two acrocentrics, which happens quite frequently, and it was already known that has an advantage over its acrocentric pairing partners And Art Daniel then pointed out that actually consistent with this, with our proposal, that actually that there is a loss in male fertility. So this crazy idea. I talked about this story last year at Matt Messelson's 88th birthday symposium, where the students basically got together, and so I told this story there. And Matt said, "Oh yeah, well, we never actually followed up and went to the fertility clinics. We never actually got funded for it, but anyway, but he had the right idea." Actually, a lot of support for this model has come, for example, in mice from Mike Lamson and Ben Black, where they showed that the predictions hold for in female meiosis, that there's expanded satellite repeats, that they get extra SEMP-A, and they make it to the asymmetric spindle, and so drive is pretty well accepted. But it doesn't stop there. Jin Chen has introduced the term mitotic drive. So we're basically talking about much later events in meiosis, but actually the very first stem cell division, there's extra SEMP-A that drives. This is in both the male germline and the female germline. And so what's happening is that all the centromeres are basically going to the stem cell, and the others and the other centromere is going to the progenitor cell. Okay? Now, why would it do this? Well, this was also explained by Jin in showing that there's unidirectional fork movement on all the chromosomes, such that all the old histones go to the stem cell and the new histones go to the progenitor cell, because the old histones go with the leading strand and the new histones to the lagging strand. So here we have a situation where a selfish process has been co-opted by the host in order to do something very fundamental for its biology. Now, developmental biologists tend to think of development, they tell us that it's very complicated. But actually, if you think about it, what this means is that the very first step in stem cell division, very first step in a differentiation lineage, is actually one that is simple chromosome mechanics. And I think it's quite interesting, and Larry would have liked this, that actually this was all discovered as chromosome mechanics in Drosophila. It's nice to see that Joe Gall finally has found an interesting model organism. Okay, but the point I want to make here is that centromere drive means that centromeres are defined by sequence. And so what is it about sequence that is so important? So we start on a new model organism, human, and this is work of Jitendra Thakur in the lab. And we did CHIP for SEMP-A. We isolated satellites. We clustered similar sequences or rank ordered based on size. And then we found the most abundant clusters. And this told us that human centromeres are dominated by two dimeric units. One that is found in centromere 1 and several others, of 340 base pair dimers, and one cent13 of 342 base pairs. Now, this was nothing new. Actually, Ivan Alexandrov had shown this way back, and he had shown it just by cutting out bands from after cutting with EcoR1, and had worked all this out. So there's superchromosomal family 1, 2, and 3. And what all three of these have in common is something called a SEMP-B box. So what is SEMP-B? Which I showed you actually cytologically basically labels all human alpha satellite. So it's highly conserved invertebrates. It binds to the SEMP-B box, and it bends DNA 60 degrees. So hold that thought. It's not essential. It's required for human artificial chromosome formations. It's found in all human centromeres except for the Y. So what we decided to do was to apply cut and run to SEMP-A and SEMP-B and see if they're in the same particle, and indeed they are. So this shows an example from centromere 7, an array that is very well studied, and you're looking at a sample of the array. And what is kind of remarkable about this is both CEMP-A and CEMP-B are showing occupancy that differs depending on the particular repeat, even though they're so similar. So let's break this in half. And now you can see that there's a big particle here in between two largish particles, but then break it in half again. And now what you can see is that you've got the CEMP-B box, and if you look at each of the four units that we're looking at here, they're all different. However, what they have in common is on one side or the other of the CEMP-B box, you basically see a discontinuity. It suggests that maybe CEMP-B is doing something to induce something or do something to cause this large complex to form. And what might that be? Well, a clue, and then I'll just point out that this also says that even though these sequences are really very similar, sequence does matter in a very intricate, exquisite way, but which sort of makes us wonder, well, what could it possibly be going on here? So the clue for us came from talking with Bill Earnshaw at a meeting, and he was wondering, he had come up with what he called the CEMP-B paradox. And the CEMP-B paradox was that African green monkeys have a lot of alpha satellite, but he couldn't find any centromere protein B there. And he asked us, why don't you take a look and see if you can, do some genomics and see if you can see anything there. So Siva Kasanathan, when he was a graduate student in the lab, decided to take on this challenge. And so if you look at alpha satellite in great apes and old world monkeys, you find that actually Bill was quite right about that. It was controversial back 20 years ago, but it's pretty clear that only the great apes have CEMP-B boxes, not the old world monkeys. So then Siva did something interesting. He asked about foldbacks. So what he did was he ran RNA fold program, and he found a striking difference. Humans have short foldbacks, whereas monkeys have long foldbacks. And on an energy scale, this has high delta G, this has low delta G. And if you ask about the Y chromosome, which lacks a CEMP-B box, it actually is similar to monkeys. It has strong foldbacks. And then it turns out that neocentromeres also have high predicted dyad symmetry, human neocentromeres. And not just from this one, this foldback, but also using another algorithm for stress-induced structural transitions, what we see is that neocentromeres are very high with respect to their DNA melting, their predicted DNA melting behavior, as well as cruciform extension. And then chicken neocentromeres, which are quite common, turn out to be off the chart. So we have a commonality of these foldbacks. Okay, what about mice? Mice have two satellites. They have a major satellite, so it's about 10% of the genome, and then there's a minor satellite where the functional centromere is. And they also follow this rule because the minor satellite has CEMP-B, and it has weak foldback potential, whereas the major satellite has no CEMP-B boxes, and it's more stable. In fact, if we look throughout the eukaryotic kingdom, we see this divergence. So, for example, corn has more stable predicted secondary structure, whereas Arabidopsis is less stable. Now, you'll see that even fish and yeast this holds for, but not only fish and yeast, but budding yeast, the same thing. You see that centromeres, the centromeric sequences, are enriched for these predicted, high predicted foldback structures. Now, in the case of budding yeast, we have different species. So we have species that are strictly defined as being Saccharomyces, and then we have others that are loosely defined. And it turns out that CDE1 that I told you about before has a different protein there. It's not CBF1, but rather we see the motif that's unmistakable because it's the one for REB1. And what we see is that there's lower foldback. So this is CDE1, and right next to it is this 80 base pair region, and what we find is that it has low potential for foldback in these species here. So what's interesting about this is REB1 bends DNA 60 degrees, exactly as I pointed out for CEMP-B. So throughout the eukaryotic kingdom, there are two types of centromeres. There are those with a DNA bending factor, and there are those without but predict strong cruciform structures. Okay, all this is hypothetical theory. Is there any evidence that this is actually the case? Experimental evidence. So SIVA looked at data that came from Dave Levin's group, where they had used permanganate-dependent nuclease footprinting. So what that is potassium permanganate will derivatize single-strand DNA, and so it won't reanneal on itself. So therefore, it can be chewed up by S1 nuclease, and then it's a matter of just sequencing the ends, and you can deduce therefore where the non-B DNA was, or you might think of as just the single-stranded regions in the genome. And turned out that half of their signal was over alpha satellite in human B cells and also over major and minor in mouse. Now, SIVA also looked at their data for resting B cells, and you can see that there's a big difference. In resting B cells, you don't really see this. So what this implies is that when you're resting B cell, you don't need a centromere. However, you can induce, when you induce proliferation, when you activate the B cells, then maybe these potential cruciforms would basically pop out, and it would be picked up by this assay. Okay, so this leads to the following speculative model. What we imagine is that there are two types of centromeres. There are those that can form these cruciforms spontaneously, and then there are those that require a DNA bending factor in order to be induced. We don't know how that might happen. It might be torsional or whatever, but basically you end up with a cruciform structure. What's interesting about a cruciform structure for centromere biologists is that the dedicated chaperone for putting in CEMP-A is called HGER. It gets its name from the fact that it was first described as holiday junction recognizing protein because it recognizes a four-way junction, and these four-way junctions by this model will be created all the time at centromeres, and therefore it would just drop its payload in order to put in the CEMP-A. So that would mean that centromere specification is really these fold-back structures, and since the delta Gs can vary quite a bit, we have actually exquisite differences you can imagine between the stability of centromeres. But I put in a slide for Ginger. Oh, I left it out. But anyway, I thought I put it in. Okay, sorry about that, Ginger. But there's another model. I think I have it here. Yeah. So another possibility fitting with what Ginger was talking about is that there's centromeric RNA that is actually, so what's happening is Reb1 we know can open up chromatin, basically open up, evict nucleosomes, et cetera. So it makes a hole for DNA, for RNA, for transcription to occur. So this is yet another model, and these are not mutually exclusive. We actually suspect that both are going on. Okay, so the work I described was that of Siva Kasanathan and G2 Thacker, and I'll stop there, and I don't know if there's time for questions. Good? Okay.